Good morning, everyone, and welcome to the final instalment of our 2023 CEBRA webinar series, or as we like to call them, the CEBRAnars. I can't believe we're at the end of 2023, or almost already. My name is Richard Kane. I'm the Principal Director of the Innovation, Research and Education branch in the Biosecurity Strategy and Reform Division of DAF. Before we kick off today, I'd like to begin by acknowledging the traditional owners of the many lands on which we meet today. I wish to acknowledge the Ngunnawal people, for me, as the traditional custodians of the land we are meeting on, and recognise any other people or families with connection to the lands of the ACT and region. Also wish to acknowledge and respect their continuing culture and contribution they make to the life of the city and the region, and noting we've got a lot of people from around Australia and indeed elsewhere, extend that recognition to the traditional custodians of all other lands on which our staff and participants are gathered here today, and indeed to all Aboriginal and Torres Strait Islander peoples attending today's CEBRAnar. Before we start, I did say it at the very beginning, but just make sure this is being recorded, so you will be able to watch it if there's any issues, and do let us know. Just while the presentation is going through by Dr. Bradhurst, please make sure you've got everything on, your microphones on mute and cameras off. As we get towards the end, Dr. Bradhurst will open it up for formal questions. You can either do that by the top of the screen there, putting your hand up virtually, or you can write in the chat field as we go along, and we will try and get through the questions as we go. So got a couple of different options there to test him. As we get into this, so for anyone who's a newcomer to any of this, the Centre of Excellence for Biosecurity Risk Analysis, or CEBRA, is a long-standing biosecurity research initiative. It plays a vital role in providing the Australian and New Zealand governments with expert biosecurity risk analysis and advice that helps inform a range of biosecurity risk management activities. Today, we're very lucky to have Dr. Richard Bradhurst, who will discuss how CEBRA has had the opportunity to validate their infectious disease simulation model against actual outbreak data, following ongoing active surveillance evidence that M. bovis has been eradicated from New Zealand's cattle population. Notice I didn't try and say that in full because I think I would struggle. Dr. Bradhurst is a Principal Research Fellow and Chief Investigator at CEBRA there, and specialises in the fusion of multiple modelling approaches to simulate the spread and control of emergency animal disease, plant and environmental pests, as well as human disease. Dr. Bradhurst is also the co-creator and principal developer of the Australian Animal Disease Spread Model, or ADIS, which is an internationally recognised leading-edge computer simulation model of emergency animal disease outbreaks, that earlier this year gained him the honour of receiving the Dr. Kim Rittman Award for Science and Innovation as part of the Australian Biosecurity Awards, and well done on that award, Dr. Bradhurst. Without further ado, I'm going to hand this over to Dr. Bradhurst to get us underway. So he's going to be sharing his screen as he goes along. Just a reminder, keep the microphones on mute and cameras off. And at the end, we'll come back to any questions that people have. So over to you, Dr. Bradhurst.
Thank you very much, Rich. And I've been looking forward to this presentation. There's nothing more stressful than doing 10-minute, 15-minute talks. So we've got a nice opportunity for a fireside chat here about potentially an obscure topic, but thank you very much to everyone that's attended. Now I'm just going to share.
Perfect, can see
All
that.
Right.
Thanks.
Yeah, so again, thanks so much for coming, and I've been looking forward to because I really enjoyed this project, and I've been doing project work for 35 years in all different sectors, and I've found that the ones I look back on most fondly, and I think that were most successful, was where the customer was engaged in the project. And this is one of those projects with MPI. So it was a University of Melbourne MPI joint project 2021-22. And our goal was to develop a tool to help MPI assess their background surveillance program in support of investigating absence of M. bovis, Mycoplasma bovis, and also to allow the tool to be of use in the future. So flexibility in the tool. And this was the project team. So CEBRA, the Melbourne Veterinary School, MPI, and our external independent reviewer was John Morton, and he did such an excellent job. I've added him into the project team without even asking him. And a shout out to Amy Burrows and Alex Crosby, because we were working jowl by jowl with them on this project, and it was wonderful. So a quick primer on M. bovis. It's a bacterial disease of cattle. It has very low clinical prevalence. So it may or may not cause mastitis, arthritis, pneumonia, abortion, middle ear issues, lameness, failure to thrive in calves, often not responsive to antibiotics, no effective vaccine. It spreads primarily from nose to nose, direct contact. So for example, nasal secretions or through colostrum or milk. Can also spread indirectly, for example, within a farm via shared milking equipment. It's not zoonotic, so the meat and milk can safely enter the food chain. Now, I shamelessly stole the content of this slide from a presentation that Amy did in 2022, but it's a nice graphic. So index case of M. bovis outbreak declared in 2017. And a decision was made to eradicate commencing in May 2018. So you can see it's not an epi curve, it's more like a case curve, just showing the number of cases. And I think the current one is just a little bit further than that, so there might be one case outstanding. Now, some definitions, and there might be howls of derision because I just sort of came up with some of these definitions based on what I've observed. But I would call passive or general surveillance more of a casual affair, so an opportunistic report from a not necessarily professional observer, maybe a professional observer, but the sensitivity and specificity of such an observer is variable. So it could be the general public, for example, just noticing some scary ants in their backyard, or a farmer noticing there's something a bit dodgy with one of the animals. Active surveillance, on the other hand, I thought of 4 types. So early detection, where there's no indication of an outbreak, so pre-incursion, and there's a system to sort of get a heads up and perhaps detect a potential index case early. So example for that might be a trapping grid looking for invasive species like a fruit fly. Delimiting is during the outbreak, where we're trying to identify the boundaries of the incursion. So that could be a report of clinical disease. It could be a contact trace that's highlighted a premises of interest. It could just be an adjoining property. So looking at places where we're kind of half expecting to find infection. And then after an infection, we have post-outbreak, where we're doing a systematic study and supported proof of freedom. And then slightly different, it was background. So there we're looking at a regimen where there's not necessarily likely to be infection, but there may well be. So it's a system to support absence. Now, MPI have been undertaking a systematic background surveillance program for M. bovis, comprising serological and bulk tank milk testing. And how were we going to help them assess the sensitivity of this system? So naturally, we go to my favourite thing, which is the ADIS, Australian Animal Disease Spread Model. And I'm going to just indulge in a few minutes in case there's anyone watching that's not familiar with it. So the next 2 minutes are, of course, my favourite 2 minutes in the day where I describe ADIS. So it's an agent-based model. It allows a user to simulate the incursion of an exotic pathogen to any farm in the country at any time of the year. It simulates a variety of spread mechanisms, direct contacts, indirect contacts, vector-borne spread, etc. It simulates detection, response options, for example, stamping out, vaccination, quarantines, contact tracing. And it also supports post-outbreak issues such as surveillance in support of proof of freedom. So that was developed with a very close collaboration with the Department of Agriculture, Fisheries and Forestry, dating back to 2012 to 2015. Test case disease was FMD, and since then it's been extended to a range of other diseases and pests. So this is just a quick movie of ADIS running. So it says a cacophony of sound, light and smell. It's dazzling, like what's going on. So let's just slow that down. In this scenario, I've released a virus in a particular farm and frozen the screen. So at this point, we're getting insight into the infection network. So a red arrow means that a infectious farm has had a relationship with a susceptible farm through an animal movement. A yellow arrow is where a hot farm and a cold farm have had a relationship through an indirect contact. The colours on the screen are the different control designations. So there might be reports of clinical disease. There could be contact tracing has yielded suspicious premises of interest. There's quarantine information. So that's our emergent behaviour of the agent-based model. That's our outbreak snapshot. This is portraying the known infection, which only the model knows. Sorry, I've said that completely wrong. It's portraying the actual infection, which only the outbreak knows, versus the known infection. So the orange is the underlying actual outbreak, and then the red is the declared or the known places of infection. That's an epi curve giving the number of new cases per day. Some monitors giving us insight into whether our resourcing, so for example, veterinarian teams or vaccine doses are up to task at this point. These monitors are very interesting. That tells us if the surveillance teams are being overwhelmed. So the fact that I'm seeing little red bars there in the Q1 and Q2 positions, that tells me that at that point, we don't have sufficient surveillance teams to respond to a report of clinical disease in a spot where we didn't expect it. So that's a high priority. And we didn't have a surveillance team to respond to a dangerous contact tracing. So red is bad there. It's a stochastic model. It means it honours uncertainty, heterogeneity. So it allows life. It allows zigzagging. And because of that, we have to run a particular outbreak scenario many, many times. And over that time, we derive the most likely outcome for that particular scenario. So there's distributions of two example outcome variables: outbreak duration and outbreak size in terms of number of declared infected farms. And we also cost every action, keep a tally of cost of control. So why do we do this? It's very complicated to build models such as ADIS. There's a lot of detail, they need a lot of data. But once you build them, you can do a lot of interesting and useful experiments without having to reformulate the model. So it's a user-driven tool in the same way as a flight simulator. Examples of specific questions we can ask: what if we have illegal movements? People don't obey quarantines. What if we have a tracing system that's not quite working well? So we miss tracing premises of interest. For our vaccination policy, is it going to be enough vaccine to cover it? Should we consider selective vaccination of only certain types of herds or certain species? Now, since the FMD model, we've expanded to other diseases in Australia. Of interest to this talk today, we've looked at M. bovis and FMD in New Zealand, blue tongue in the US, FMD in Canada. In Europe, a bunch of diseases: PPR, sheep goat pox, classical swine fever. Perhaps at a tangent, we adapted the model to invasive species. We looked at yellow crazy ant and oriental fruit fly, and currently looking at capra beetle. And there's a human variant of ADIS as well, which looked at rotavirus. So back to our task at hand, modelling M. bovis. So if we can break it down, what do we need to do in the ADIS model? We want to simulate the NZ cattle population in time and space. We want to be able to introduce seed infection into any farm. We want to be able to simulate spread between farms of M. bovis and spread within a farm, and also be able to simulate background surveillance, comprising serological bulk tank milk testing. So our high-level strategy was, let's assume that it's been eradicated. So we initialise the population as naive. We seed a single case of M. bovis, allow it to spread. We can currently operate the background surveillance regimen, and we just simply see how long it takes for our scheduled surveillance visits to pick up that outbreak. And when it does pick it up, that's our time to detection. So we repeat that systematically for a broad range of seeding scenarios across New Zealand. But how do we represent the cattle population? It's an agent-based model, so each herd is an agent. A herd is just simply a group of animals, same species, same production system, same management system. A farm is a container for herds, so a beef sheep farm would have a sheep herd and a beef herd. So these are just abstractions on how we view the population. An agent can have static attributes such as the type of farm, on-farm biosecurity measures, and dynamic attributes. So in acquiring infection, whether we have clinical disease, and if and when control designations are applied to a property. So that was a New Zealand herd population coming in at about 57,000 farms, and each farm individually represented, so you can interact with each farm. Each farm has its own story, but we also categorise farms as farm types, and that's useful for modelling purposes, just to have to assign behaviours that aren't necessarily obvious to an individual farm, but they're more applicable to the type of farm, the production system. So I'm not going to labour this, but there's a mathematical system inside each herd. So we have 57,000 OD systems sitting there waiting. But it's much easier just to think about this in terms of colourful boxes. So compartmental model is a way of looking at a herd and just thinking about the proportions of the herd that are in a certain classification. So S is susceptible, E is exposed to a pathogen, I is infectious, R is recovered, D is deceased. And just as a quick quasi-animation, which is actually me just flicking, I'm loading 4 susceptible individuals into the S compartment. I challenge one of them with a pathogen, they become exposed. A little bit later, at the once the latent period is expired, they become infectious, in which case they can have a relationship with the other susceptibles through that beta I pathway. And so sure enough, another chap gets exposed to the pathogen, and one by one they're all exposed and eventually become infectious. Then clinical signs kick in for one of them, so they're not feeling well. So he appears in both the clinical box and the infectious box. So the system models clinical disease quite separately to infection, just for convenience. The rest of the exposed chaps become infectious, and then at a certain time they recover. They go. So we've got no mortality in this system, so none ended up in the D compartment. They've all ended up in the recovered department. So at this point, they would have degrees of natural immunity to the disease. Now, if we just had one farm in the country, that is our model. It is just that OD system predicting prevalence over time. So on the right is a graphic of the one herd. And the little white ball is just showing that over time we have different ratios, proportions of that herd in different compartments. So the red curve is giving us indication of portion of the herd that is infected, so either exposed or infectious. The brown curve is serum prevalence, so that's the proportion of the animals that have had an active immune response to bovis. And we derive that from the infected prevalence, but we take into consideration the average seroconversion characteristics. So by that, I mean the proportion of the herd that will seroconvert, the time that they'll take to seroconvert, and the period that they'll be seropositive. The final prevalence curve of interest is the cream curve, and that's called lactoprevalence. Now, we originally called this milky prevalence, but we felt that lactoprevalence had more street credibility in the scientific community, and I think someone was worried that milky prevalence sounded a bit like a hipster criteria for ordering a latte. So we're much more comfortable with lactoprevalence now. So that reflects an estimation of antibodies expressed by seropositive cattle into the bulk tank milk. So we derive that from the serum prevalence, but the trouble is it's a bit more vague because we have cows that are both seropositive and seronegative, contributing milk to the tank, so there's a dampening effect. There are observed seasonal variations in the level of antibodies that cows express into the milk. And so we add noise. So that's the wiggly wobbly part of that cream curve, is the dampening and the addition of seasonal fluctuations and noise. Now, ADIS provides a number of mechanisms for herds to have a relationship. So for an infectious herd to somehow influence a susceptible herd. Now, in the case of M. bovis, we're only interested in 2 of those. So the rest of them are disabled. We want to be able to simulate animal movements between farms, direct spread. And we want to be able to simulate what I mentioned before, that there might be nose-to-nose contact across a fence between neighbours. There could be strays, some sort of mystery relationship, if you like, that an adjoining farm lights up hot, and we're not immediately sure why. You know, there's been no animal movement between the farms. So that's more a proximity-based or a spatial kernel-based method of spread. So firstly, our direct spread pathway. And we were very lucky. We're able to leverage off some excellent work done by other colleagues. So there was a project in 2021, which I think it was called the Social Network Analysis Project, and it characterised cattle movements, so mining the NATE database and looking at movements by TLA community. So that was by Mason et al. So we used that to study, and then we supplemented that with some additional work from MPI and some work by Van Andel and colleagues in 2016. So what that enabled us to do was to characterise in the terms that ADIS needs to generate direct contacts, the likelihood of animal movements between farms. So we consider the frequency of movements off a farm, the size of consignment, potential distances of movements, destination types, and the direct pathway allows us to simulate these direct movements. And we add stochasticity because not every direct movement will result in infection. So you can have an infected farm that's just lightly infected, and you might be lucky enough just to consign animals that are free of disease. So we take into account the level of prevalence inside the infected farm, and that's where our ODE system feeds that information into the agent-based model. And what this does is it enables us to generate distributions of infection networks. So ADIS can just replay historical animal movements, so you get the actual network, or we can be a bit more colourful and use the stochastic method where we generate a whole range of plausible infection networks. And that's useful because life is variable and uncertain. So if we can test a particular control strategy against a range of possible outbreaks, a range of infection networks, gives us more confidence in how that control policy is shaping up. Now, our local spread pathway, as I mentioned before, is just a spatial kernel, which is a pretty standard technique of conveying how likelihood or danger is increased the closer you are to the source of the danger, and then it decays in a certain shape the further away you get from that hot farm or the source of danger. So you can choose different shapes, and I've just got a FYI graphic there showing how you can choose different rates of decay of risk, if you like, with distance. Now we ought to be able to start infections, so we use our standard seeding mechanisms at ADIS. We can just pick on a particular herd, which is what I did in that little movie that I played. Or you can do random seeding, whereby you sweep infection broadly. You can choose different herd types, different herd sizes, different regions. So for example, let's just pick on all large dairies of a certain size in a certain region in a certain month. Once we've defined our primary case, we allow the direct spread, local spread, and background surveillance components to operate concurrently. ADIS is a highly concurrent piece of software. There's something like 40, 45 concurrent threads. Or you can think of that as programs, means of execution, all cooperating. So it's an extremely complex bit of software. Now, one of the neat things was, because we had outbreak data, we were able to do some nice validation studies. So in this example, we introduced infection into roughly the area that it's thought that M. bovis first appeared in, roughly the region and the time of year. And we allowed it to spread up until detection. So we did a sweeping process. We seeded in that area of worry and just let the spread pathways and the background surveillance collide. And we ended up with a heat map, which is a picture of New Zealand on the left, generated by ADIS. And on the right, we've got the actual cases. So there was a very nice correlation, and you can see spatially, if you squint, it correlates quite nicely, but also more analytically, it was consistent with respect to the number of infected properties, the mix between beef and dairy properties, and impacted communities, so regionally. So that was a very nice opportunity to be able to do that as one of our tests. Now, a bit more information on the background surveillance component. It's quite simple, and we simply just play a script. So the picture on the top right is a text file, which just dictates what surveillance visits to conduct. So we have a calendar date, a herd identifier, the type of herd, a test, and a sample size. And then we look up the details of the test to conduct in the table below that. So, for example, in the first entry on the 1st of January 2022, I'm going to go to herd 43420, which is herd type 6. So I go down and I see, oh, that's a large beef commercial. So I'm going to run a CELISA serological test, sample size 100. I'm going to look at the sensitivity of 0.6622 and specificity of 0.9988. So that's just made up data. I haven't given anything away there. So that's just to show how it works, but it's quite a simple, elegant mechanism. And it puts the power into the user, because they can define how they want to populate the surveillance input file, with different regimens with no programmatic input. Now, I'm not going to dwell on this too much, but because it's a simulation model, we're actually, in effect, going out to a farm and conducting the test inside the model. And so the way that we determine whether or not we find infection or not depends on the prevalence curves inside the herd, which the model is generating. So we know if the herd is uninfected. We know if it's slightly infected, or if it's deeply infected. So that is revealed to us by that P of T term, which is serum prevalence as a function of time. We take into account the sensitivity of the test, the number of animals in the sample, and that gives us a probability that it's a true positive, which we then construct a binomial distribution to sample from that, and we get a yes/no answer as to whether it was a true positive. In a similar vein, we determined if it was a false positive by looking at the specificity of the test, the number of animals sampled. Similarly for bulk tank milk testing, except that we just have one sample because we're testing the tank. We're not testing individual cows at that point. And relies on the lacto prevalence, and you can see there an artifact that it used to be milky prevalence, and that I've got M of T. I should really have L of T there. Well, and similarly for if it's a false positive in bulk tank milk testing. So with those four little equations, enables us to make a determination for each surveillance visit that we simulate. And here's a movie of the model running. So the little somewhat creepy flashing yellow dots, it looks like some sort of bacterial issue, but that's the surveillance visits being enacted on farms across New Zealand. This model runs quite quickly, so that's why it's just a flash and it goes on to the next surveillance visit. And then in parallel, if you look closely, you'll see the red arrows. So that's the infection network coming into action in response to a seeding in a certain area. And we allow the direct spread and the local spread pathways and the background surveillance components to collide. And when they collide, at some point we get our detection. And that's where we get a score in the blue histogram, which is the days to detection outcome variable. And the red histogram is the number of IP, or a number of infected farms in play on that day of detection. So that's the model running. And ADIS produces a alarming amount of output variables. But for this project, we were interested in specifically to be able to go back and find out details on every single simulated surveillance visit, so where it was, when it was, the type of test, the sample size. We didn't do costs in this project, but you can assign costs to actions, and then so part of your post-simulation analysis would involve perhaps around relative comparisons of cost effectiveness, but we didn't do that for this. We record the test result, true, false, positive, negative, within her prevalence levels. So gives us an opportunity to do forensics on how well our tests are performing, because we secretly know what those serum prevalence and lacto prevalence and infected prevalence levels were on the day that we locked up to do the test. Time to first detection, where it was detected, and what was going on outbreak-wise on that day of first detection. So whether we just got in and it was a massive outbreak or just a small outbreak. So all this is wrapped up into text file outputs for analysis. So what we did was we did a range of outbreaks. So quite an extensive set of simulations across different herd type seeds. So those 6 or 7 different herd types that I flashed up earlier. So we would choose by herd type, by region, basically just covered all of New Zealand with simulations allowing incursion of M. bovis in a naive population, so to speak. So just we initialize everything to susceptible, introduce infection, and allowed local spread, direct spread, and background surveillance to run. And then when we get a hit, that's our time to detection. So what we found was that after 2 years, we were successful in detecting outbreaks 94.2% of the time. If we kept background surveillance going for 3 years, we would detect 98.9% of outbreaks. And if we kept it going for 4 years, 99.6% of outbreaks. So that's our measure of system sensitivity, SSE. And there's a nice curve there on the right, just by region, but just showing that you do get a significant improvement in sensitivity by maintaining that program into the 3 and 4 year mark. Now, one thing that we can do with our derived estimates of system sensitivity is plug them into this formula. which gives us an estimate of the probability that M. bovis is still present in the population. So the inputs to this is the prior probability P, so estimated probability that that M. bovis is present in at least one cattle herd in the country, our system sensitivity and system specificity, which we just held as one for the purposes of this analysis. So when we use that formula across our range of simulation runs over 2, 3, and 4 years of background surveillance, we come up with different values depending on what prior probability we choose. So, and the curve there on the right shows that we are getting, again, a great benefit from heading into that, into the 3rd and 4th years of surveillance. It drops down significantly from the 2nd year. But just to bring this into something that's more digestible, if we assume a 50% prior, which is quite a conservative estimate, so 50% chance that M. bovis is still out there, we have a 94.5% probability of freedom after 2 years with no detections, a 98.9% probability of freedom after 3 years with no detections, and 99.6% probability of freedom after 4 years with no detections. And the thing that MPI liked, and I think was a great result, was that we came up with quite comparable conclusions to another study by Brendan's group, which used a completely different method, used scenario tree modelling to estimate theoretical probability of freedom. So they came up with 98.6% within a confidence interval. So, you know, broad agreement between the two modelling approaches, and I think a reasonably clear signal on the value of staying the course beyond 2 years of background surveillance and pushing into the 3 and 4 mark. So that's our project report, which sadly is not publicly available because it does sort of contain more sensitive sort of data about farm locations and characteristics of the program. So that's just an internal report. However, I'm drafting a more benign paper, really just going through the methodology that I've shared with you today. So that will be available down the road, just to capture the approach that we used, which I think is an interesting use of an epi model. And acknowledgements. So Australian government has always been great supporters of modelling and support of preparedness of emergency animal disease. They've got a rich history, 20 odd years of believing in being able to quantify opinions and estimates of what EADs may or may not do. So they've continued that support to this day. So they're strong supporters of our efforts, and they provided the ADIS model to MPI free of charge as being part of the ADIS Secret Society. So just acknowledging the department there and thanking them. And as I mentioned earlier, really great project, really good team. So MPI, it was Amy and Alex, sort of the day-to-day, and Sarah Lee and Mary, more managerial support, administrative from the MPI point of view. CEBRA was myself and Andrew. In the vet school, Simon and Mark, and our wonderful external reviewer, John. So he's part of the club, whether he wants to be or not. And if you're curious about ADIS, there's a website there that you can go to, adis.org. And please don't hesitate to get in touch if you've got any questions beyond what we can look into at this CEBRAnar. So that's me, and I think I've come in under time. So
no, you've done well, Dr. Bradhurst. That was excellent. Thank you for that excellent presentation. Now, I'm just going to throw it open. You can either put your questions in the chat field on the side there, as I said, or you can formally click on the hand button and raise your hand if you've got any questions. So I did have one that had initially come through just while we're waiting for everyone else to get that in there. This was just on the ethical considerations in terms of the data. So Dr. Bradhurst, one question was, are there any ethical considerations in using real outbreak data for simulation modelling?
Well, yeah, that's a difficult question. I mean, the sensitivities, of course, I don't know about ethical, but yeah, certainly the sensitivities that we're very much across of use when we do use actual data. So projects like that tend to be very locked down and more specific to the people that we're dealing with. And often we do modelling studies with more generic synthesized data to just be more comfortable in that space, and we can share more freely. But yes, I am cognizant of ethical issues and sensitivities, so I'm not quite sure what to say apart from that, but yes, there are sensitivities.
It's a bit of a tough one in terms of that, but I think, and Professor Robinson has just added his part there in the chat. We've got another one here for you. I'll read it out, which will give you a bit of time. It's just coming through in the chats there. What is or are the applicability of ADIS for other diseases, and where would you get the data from, noting potential sensitivities? And would that be from New Zealand, Australia, etc.?
Well, I think we're around the world, like it's, as I mentioned earlier, it's the same model code used in many different places for a range of diseases. So what we're trying to do as a development group is come up with generalized approaches. So I kind of view ADIS as a toolbox, and for example, with the lumpy skin disease model that we've just been working on, we've come up with some fresh or new methodologies on how to view mechanical vectors, which we hadn't before. We've done biological vectors with blue tongue. So as each project comes up, there's potentially different disease. There might be nuances on transmission, and that all gets folded into the core ADIS product. But it's disease dependent. So someone said, yeah, let's have a crack at equine influenza. We'd have to sort of go back to stage one and say, well, what's the data like for horses? Do we need to think about feral horses? And and hopefully we'd be able to reuse some of the techniques that we've developed for other diseases, and we might have to develop some new ones for new diseases.
Thank you, Chris. That was a bit of a curly one. I think you just touched on it there. I just had another question that had come through in terms of how has the simulation model itself incorporated lessons learned from previous outbreaks and simulations to improve its performance?
Yeah, well, we're always looking to improve. So we do validation studies in Europe using outbreak data in the plant pest space. If we have data, we feed that back to sort of better tune the techniques. But often these pathways are data driven, so it really hinges on having the data to start with. And if we don't have data, there's an option to switch just to more analytical modeling. So you can do very fast, quick jump diffusion models using ADIS. You can turn off all the complex data-hungry pathways and just have very simple, like a rate of spread, in a topology of vulnerability, if you like. So you can have selective diffusion. And you can perturbate a system with jumps just to replicate that. Sometimes you get satellite clusters of infection or outbreaks.
Excellent. And it's just in the chat field there. This is from Jess. Will ADIS expand more into human disease outbreaks over time? Thank you.
Yeah, look, I pondered that because when we did the rotavirus study, I got all giddy, and I thought, yeah, this will open up zoonosis and more human disease. But then the human modelling space is really filled with a lot of clever, competent, well-funded bodies. So they didn't need another body in that space. So I've kind of backed away from that human side. But the functionality is there. So the same way that I can declare a farm to be an agent, I can declare a human to be an agent in ADIS, and I spread disease inside the human mathematically, and then I can spread disease between humans in an agent-based manner. But at the moment, that's just off to the side. But who knows? One day we might consider melding that functionality in with an animal disease model and think about zoonosis more seriously. But it would have to, maybe a PhD student would put their hand up and run with that over a 3-year period. That would be a nice outcome.
There you go. To anyone listening, that was a free idea there from Dr. Bradhurst. Everyone's being shy. They're not putting their hand up and putting the camera on. So that's okay. You can come through the chat. That's fine. Curly one from, must be a Professor Robinson here. What effect would uncertainty in farm occupancy have on ADIS projections? Is it likely to be quite robust? Would it be feasible to expand the model to account for that source of uncertainty?
So by farm occupancy, do you mean like understanding the location and nature of the farm? Andrew?
I'll go out on a limb and turn on my microphone and camera. Richard, what I mean by that is the population of animals on the farm, both the composition and the size of the population on the farm.
Yeah, so that's a good question. And there has been studies using ADIS in New Zealand in FMD 2016, where Mary actually compared a data set that was comprised of actual sizes, census driven, versus a data set that just used average farm sizes. And from memory came to the conclusion that it was worthwhile having the actual data. Like you got quite different results between the two. So yeah, I think there's probably scope to do more work like that, because that's always our biggest Achilles heel, if you like, that data quickly becomes smelly, out of date, movement data, farm production style data, and carrying capacities of farms, and even if the farms are still there, it's in a state of flux. So I think there's an opportunity there just to get a better handle on how much we're relying on accurate data. And if it turns out that it's not that important, then awesome, we just use synthesized data and go with the trends. And it's a bit like, with the COVID modeling, do you actually need to model every specific individual and their particular personal contact network and where they go? And that would, of course, be impossible. Or do you just generalize, and you look at a demographic bracket and say, yeah, that type of person's probably got this style of contacts or these other style of brackets, and you just abstract, and that's perhaps all you need to do. But I think in the animal world, because we have things like NILS and NATE where we do collect data, there's a strong perception that we should be making full use of that and better use of it. But yeah, uncertainty in general, ADIS is like highly stochastic, so we allow stuff to go funky. Like transmission rates, you can allow them to vary, farm sizes, whether or not contacts occur, whether or not control measures are heeded, whether or not you have resources, all wibbly wobbly stuff, that's in there, and it's up to you as to whether you turn it on or off or up or down.
Excellent. Thank you. And thank you, Professor Robinson, for turning your camera on as well. Now, someone's put their hand up and I have it as Andrew Breed. So Andrew, fire away, mate.
Thanks, Rich. Yeah, thanks, Richard, for a really great presentation. It's always so reassuring to see when some kind of validation activities can be undertaken for modelling use. My question is, I guess, following on, I think, from a couple of others, looking at the sort of what's happened in the human health area and increasing use of models, I guess, since COVID for use during outbreak response. When I think historically, at least in the animal health area, we've mainly used models quite deliberately sort of for preparedness. I was wondering how you see things, I guess, kind of from where you sit in terms of the use of models in the animal health sector during outbreak responses. Thanks.
Yeah, well, look, I would totally cede opinions to the people doing the job. So people in the war rooms think that models can be of use, then, you know, I'd be delighted to provide those models and be of use. But you're right, up until this point, like a complicated simulation model like ADIS has been more geared towards preparedness, where you've got time to assemble the underlying data, and it is, you know, rich in data, and you've got time to run creative scenarios and try and ascertain messages that the emergent behaviour is hinting to you. For example, vaccination in some parts of Australia makes a huge difference in an FMD outbreak. The model's suggesting that. Other parts of Australia, other times of year, it makes no difference. And, you know, it's kind of hard just to intuit that. And very hard to defend an opinion like that. But, you know, the model helps quantify areas, spatially and temporally, where the heterogeneity matters, and that it's not just the one solution for policy. But yeah, I think you're right. That's a good comment, that modelling is certainly in the public eye more, and the terminology is around the dinner table. And certainly people think I'm a lot less weird now because they know they've seen people on TV talking about similar concepts. So that's one good aspect. But I would like to think that ADIS, you know, would be of use during response, but it would, of course, be fully determined by the people doing the response, you know, whether it can help them address certain questions. Last thing you want to do is give a model to an academic and get them to make a call on something.
Yeah, thanks, Richard. I think those are good perspectives. I mean, yeah, my feeling is that, yeah, since COVID, yeah, modelling, not only do people think that modelers are not quite so weird, but they're thought of as being, you know, something you can kind of turn on the tap and just get the outputs and are going to use them. And so, yeah, I think it's an area that we need to get better organised in, I suppose, in terms of which models are we going to use, and what questions are they going to help us answer. Thanks.
And that's a good point related to that. It's these models are seductive. You know, they look like they know, and you want to believe them because there's lines definitively going between properties, and that is an illusion. You know, what the strength is the distribution of plausible outcomes that it can produce. It's not telling you which farm's lighting up tomorrow, but it might tell you, you know, the potential magnitude or spatial extent, or the, you know, resourcing requirements. So I think it's an important message that needs to be conveyed as to what they are useful for and what they're not useful for.
Thanks, Andrew. That was a good question and good, you know, example there of how ADIS could be used for response activities, or at least considering some of these things. We've got, don't forget, always people moving into different jobs, different spaces, so you can lose that corporate memory and understanding of how to manage those sorts of things. So I think Richard has posed some really good questions for further consideration in that space. I can't see anything else coming through, but just noting the time of where we're at. Any last questions, either in the chat field or by putting your hand up? Otherwise, I'm going to try and close it on time, which is unusual for me. Gave it that 7 seconds of silence. I can't see anything there, but Dr. Bradhurst, thank you very much. That was an excellent way to end our 2023 CEBRAnar series. So much appreciated for all of that and taking the time to go through some of those questions as well. For everyone else that was here, it's being recorded, so we'll be able to play it back, and happy to share it on to others if there is any interest. Details on our 2024 CEBRAnar series will be announced early in the new year. We'd like everyone to get a bit of rest over Christmas and into the new year. So we will get some info out probably later in January about that. And to be the first to hear about it, you can follow through our research and innovation section. And we're now on Humanitics, I think it is the platform that we're using for promoting all of this work. But thank you very much to everyone. Thank you, Dr. Bradhurst, again, and the CEBRA team. It was excellent, and wishing you all a very early Merry Christmas and Happy New Year. Thank you all.